not a morphologist, but a biochemist. He discovered lysosomes as, or predicted their existence as organelles based on his studies discovering that there were enzymes within cells that had were most active at or optimally active at an acidic pH. So he predicted the existence of lysosomes, coined the term, and then, of course, there were many questions about what such organelles would do, and this illustration here is simply one of the early views that lysosomes could lysosomes could rupture and release their organelles, and as a result could damage cells. I'm having a little bit of a technical problem here, and I'm going to have to close my screen just for a second. Siobhan, could you just give me feedback that because I've lost the Citrix material on my slide? Am I still broadcasting here?
Yes, Steve, I do see you here. I see a slide that says the discovery of lysosomes, 1955 at the top.
Yes, okay, so you can hear me and everything is fine. I'm not getting any feedback, so I was uncertain.
Yes, everything looks great so far.
Okay.
Thanks.
So, so that was Deduve's contribution. Of course, he predicted the existence of these organelles, but he was not the morphologist who actually discovered them. This really went to Alex Novikov, who was working here at the Albert Einstein College of Medicine, who was doing electron microscopy in the late 1950s, 1960s, and using acid phosphatase histochemistry actually demonstrated the existence of these organelles containing enzymes with acid optima within cells. And Novikov really deserves a great deal of credit for this real morphological identification of what Deduve had proposed existed. And of course, as I mentioned earlier, we're now at the 50th anniversary of the articulation of the lysosomal disease concept. This was by H.G. Harris, who was working with Deduve, or down the hall from Deduve, and was analyzing tissues from a patient with Pompe disease, a glycogen storage disease, and discovered that there was an absence in activity of the alpha-glucosidase enzyme, which had been discovered to be a lysosomal enzyme. And really remarkably, Harris went on not only to say, well, Pompe disease must be a disease in which a lysosomal enzyme is deficient, but he went on to say, well, all of these other so-called storage diseases that had been known for many, many decades, really starting at the beginning of the early 1900s, could also be due to defects in other lysosomal proteins. Really remarkable insight into a whole family of diseases, and Harris deserves really great credit for articulating that idea. And a lot of what was going on subsequent to the discovery by Deduve and by Harris sort of came together in 1973 with the publication of this book, Lysosomes and Storage Diseases. Really, truly remarkable text that I recommend even junior and senior people go and find a copy of. It is still available as used copies. And really, again, remarkable insights into where the field was at that time. And within this, the different chapters in this this book, they cover many different types of topics. And I'm going to use, actually listed many here, some of which are included in their text and some of which are not, but it's, I think it makes a good starting point in terms of discussion about the sort of modern state of what we know about lysosomal diseases. And the first of these is lysosomal proteins implicated in disease. And here's a statement out of that book by Harris, really showing his knowledge that lysosomal enzyme deficiencies would be key, and at the time there were 40 of these by 1973 that had been documented, and that this would be the key to understanding an individual type of storage disease. But you'll notice there he also mentions or structural proteins, which are badly known. And again, real insight, because at the time there really wasn't much known about lysosomal structural proteins that could in fact cause disease. And the diseases that are discussed within that book are all lysosomal enzyme deficiencies, and I've listed them here. And but even that list, considering it's 1973, is really quite impressive. But of course, as the years have gone by, that number has grown dramatically. There are now over 30 different lysosomal diseases that are known to be caused by lysosomal enzyme deficiencies. And there are really a host of other diseases as well, lysosomal diseases caused by enzyme deficiencies where the enzymes are not in the lysosome, they're in the Golgi or the ER, or by proteins that are soluble to the lysosome and have other functions other than enzymatic activity. There are also transmembrane proteins of the lysosomal membrane that can be defective. And the purpose of this talk is not to go through all of those individual diseases, but just to lay the groundwork for understanding that this is a very complex family of diseases, and they're caused by defects in proteins that are critical to the function of the lysosome. And so we now recognize something almost 60 different lysosomal diseases, and their molecular bases are really quite diverse. And I've added a little note here about animal models. I think it's unlike other fields of neurodegenerative disease like Parkinson's and Alzheimer's, which for so long had little or no animal models. The lysosomal disease field is really quite different. We've had both rodent and large animal models, that is lysosomal diseases in dogs and cats, also in livestock like sheep and cattle. For many, many, many years they've been recognized, and many of them have been absolutely key to understanding different aspects of the pathogenesis of these diseases. They are remarkably good at replicating the events that we know occur in the human brain and other human tissues as a result of a given lysosomal protein defects. So and again, these animal models have really added to our sense of the complexity of these diseases. And we can make a simple diagram, as is shown here, where, of course, you've got an initial gene defect, a single, in many cases, single protein defect, and then, of course, lysosomal storage occurring, this expanding into impacting other aspects of events within cells. And of course, in terms of disease, and my own focus, of course, is on neurological disease, we sort of can describe this as an expanding cascade of events. But this kind of diagram is really rather sad when you really think about the complexity of the disease itself. So this one is a little bit more detailed, and it's listing the various types of lysosomal diseases that we know, and the enzyme or other protein defects that are known to be associated with Gaucher or GM1 gangliosidosis and so forth. But of course, what we're trying to do as we try to understand these diseases is really understand, for an individual disease like mucolipidosis type 4, for example, we're really trying to take apart and understand all of the cellular abnormalities that are generated as a result of deficiency in mucolipin-1 or for beta-galactosidase and GM1 gangliosidosis and so forth. And what you can see by just the way these arrows are displayed here in terms of the cellular phenotypes, that there are really two different kinds of pathology that are really here. One is divergent in the sense that in a given lysosomal disease, there are many, many different characterized cellular phenotypes that result from that. You know, everything from storage and axon hillock enlargement, which is known as meganeurite formation, to changes in axons and myelin and so on and so on. You know, you can refer to that as the divergent pathology of an individual disease. But the other really important feature about this when you're comparing different lysosomal diseases is there's this tremendous convergence where so many lysosomal diseases, in spite of the dramatic difference in their original protein, gene and protein defect, actually are remarkably similar at the cellular level. And sometimes they're remarkably different, but through those comparisons we can learn a great deal about, ultimately, about the importance of that initial protein that was responsible for the disease itself. And of course then the other thing we want to do in this is be able to take those individual cellular phenotypes and understand how they individually contribute to different features of the neurological or other aspects of the disease itself. And again, my emphasis here is on the neurological side because I'm a neuroscientist studying lysosomal disease. But you could say similar things about bone and liver and so forth. But ultimately, I think this is our goal, is to understand these cellular phenotypes, their molecular underpinnings, and of course, if we're trying to translate this into therapy, into ways to intervene and ameliorate either one or all of those cellular phenotypes and then the neurological outcome that grows out of that. So another issue as we go through this list is heterogeneity of lysosomal storage. This again was a topic that Harris focused on in that 1973 text, along with other authors in that text. And of course, as the quotes here from Harris indicate, he would have predicted this on the basis of the nature of lysosomal enzymes. And of course, he's absolutely correct in all of his reasoning there. But what I'd like to sort of take you through is to argue that it's even more complex than the differential impact of an enzyme on the substrates that are being degraded. It's actually much more complex than that. And our first experience with this in terms of my lab was many years ago, looking at a model of alpha manocytosis that had been discovered in cats. And what's shown here in this opening slide is two neurons sitting side by side in the cerebral cortex of this cat, and it's a PAS stain, periodic acid Schiff reaction stain of a plastic section. But what you see is one neuron has a great deal of storage material in it, sitting right next to the nucleus. The other cell, you can barely see the nucleus, and you can sort of make out the cytoplasm around it, which looks rather foamy. And the reality is both of these neurons are undergoing storage, but one of them, the one that has PAS positive material in it, actually has a completely different kind of storage body in it than the other one. And because the nature of the storage material that's building up in one of these neurons is very different than what's building up in the other. And we know through subsequent studies that this, in fact, is reflecting the PAS positive staining. It's largely reflecting the presence of ganglioside storage, both GM2 and GM3 gangliosides, which in normal brain, normal neurons, essentially is absent, but is building up in this and other lysosomal diseases and neurons. But in this case, it's really reflecting this, this heterogeneity of storage between different neurons, and again, the ganglioside that's accumulating is really barely detectable in HPTLC type analyses, and in fact, total gangliosides are not elevated in this disease, and yet at the cellular level, there are abnormalities that are readily evident. We went on to look at this in other diseases, And again, much of this work now somewhat dated, but I think really highly relevant to our thinking about lysosomal diseases today. Here's Sanfilippo type A disease, again now in a mouse model, looking at immunostaining to heparin sulfate. And what you see in here is there appear to be areas, well, the normal neuron, first you get this very diffuse staining, but in the diseased neurons, you get dark granular staining in a lysosomal-like pattern right next to the nucleus that seems to indicate heparin sulfate proteoglycans might be accumulating there. And but then there are other areas in the cell here that are devoid of that staining, and it makes you wonder whether there's not something else also accumulating in that cell. And in fact, once again, as had been known from biochemical studies earlier, there is ganglioside storage accompanying the storage of glycosaminoglycans in a disease like Sanfilippo. In fact, the ganglioside storage is really quite substantial. And again, you can see it as both GM2 and GM3 gangliosides here building up in neurons. And here, if you take this a step further and look at it from a confocal point of view, what you find, and here we're looking both at GM2 and GM3 gangliosides as well as cholesterol, what we find is that there is, again, and this time individual cells vary a little bit. This neuron from the cerebral cortex has cholesterol accumulation accompanying the ganglioside accumulation, whereas the neuron on the right does not. But what is really remarkable here is the way the GM2 and GM3 gangliosides are largely in separate compartments in the cell. And so here is this heterogeneity of storage within individual neurons, not between neurons. And this is a pattern that has played out in every lysosomal disease we've looked at in terms of these two particular substrates that we know accumulate in quite a few different lysosomal diseases. So this is Niemann-Pick C1 and NPC2 disease, again showing really, for the most part, separate compartments for storage of those two substrates. And then here's MPS 7, very similar picture. And even if we go and we look at the presence of cholesterol and GM2 ganglioside in a disease like Niemann-Pick C, which is really thought of as a primary cholesterol storage disease by many individuals, the GM2, which abundantly accumulates in neurons in this disease, is largely accumulating separate from where the cholesterol is. And again, just indicating the complexity of this storage process and what's going on at the cell biological level in these neurons. It gets even more complicated because evidence we have indicates that storage is not really even limited to organelles that we would identify as late endosomes or lysosomes through a LAMP1 or LAMP2 stain. And this is seen here simply by doing, again, confocal microscopy and labeling with antibodies to GM2 and LAMP2 or GM1 and LAMP2. And what you find are these ganglioside-laden vesicles within the cell that, in fact, don't appear to be surrounded by a LAMP2 positive membrane. Same thing with cholesterol in Niemann-Pick C disease. Some of the cholesterol appears, again, independent of LAMP2 staining, indicating or suggesting that it's in a compartment outside of the late endosome lysosome, which, again, might not be all that surprising, but I think it probably is highly relevant to the disturbances that are being set in motion by this storage process. So I'll move on now to another topic, autophagy, and I say listed here as autophagy rediscovered because if you look again back at the Harris and Van Hoof text from 1973, autophagy is a major interest area in terms of lysosomes and lysosomal diseases in those days. Literally every illustration that is shown shows a tie-in to autophagic vacuoles. And for reasons that aren't all that clear to me, this sort of a concept of the lysosome and autophagy really went to the back burner for many decades. And it was really only in about the year 2000 and right after, and this really nice graph done by Daniel Kliansky, showing the publications and their increases on the topic of autophagy, starting about 15 years ago. And this was happening, of course, because scientists were studying the autophagy mechanisms in yeast, discovering many, many different proteins that were involved in this process, and their counterparts in the mammalian system were being identified. And so it was finally possible to take apart the molecular machinery of autophagy in mammalian cells. And everyone really got very interested in the clear significance of autophagy to cells of all types became readily apparent, even for neurons. And I remember very well when these two papers appeared in 2006 that showed if you knocked out some of these key proteins involved in the autophagic pathway and thereby blocked autophagy, that in fact it would affect even neurons. That is, that autophagy was a constitutive element even of neurons, and as a result of those knockouts, neurodegenerative disease occurred in those mice. So really a major step forward. And of course, not surprisingly, labs that were working on lysosomal diseases found this work interesting and pursued it and asked questions about the role of autophagy and possible autophagy compromise in lysosomal diseases. And this work was, there are a number of independent publications on this, but it was summarized in this Lieberman paper in 2012. And much of the emphasis here was that there appeared to be a block in autophagy mechanisms in lysosomal diseases, and that this could be really a major part of the pathogenic cascade, resulting in the presence of effete mitochondria and the consequences that could grow out of that, of toxic proteins in the cell building up, and the possibility of cell death. Now, those of you who have heard me lecture before know that one of my points that I like to stress is that neurons, in fact, don't just die with lysosomal disease, and that death is what generates neurological disease in the affected individual, but rather neurons can live in a state of compromise with lysosomal storage and its attendant issues for many months, many years, in fact. So as we think about a diagram like this, I would just emphasize the fact that these arrows are longer than are illustrated here, and then in fact cell death may be a very long-term consequence, but that dysfunction of those neurons, again, possibly tied into autophagic compromise, is a factor, but it's not leading to immediate death of those cells. We've done, actually, in my lab, a great deal of work focusing on this through the good efforts of Matthew Machaney. And here's some illustrations from his work just showing the presence of one of these macroautophagy adapter proteins, P62, the accumulation of which is often cited as evidence of block in autophagy. In fact, it was cited as such in the review paper I just mentioned. But it's really intriguing to look at different lysosomal diseases and the extent to which P62 accumulates in these aggregates in these different diseases, not always in exactly the same pattern or in the same cell types, but clearly is a factor throughout. And it really raises a lot of questions about whether the P62 is simply building up as a result of a compromise in autophagy, or whether there are other issues going on as well. And I'll return to this topic a little bit later in this discussion. The other, of course, major aspect here in dealing with the sort of modern view of autophagy and lysosomes is that there are so many different organelles now within cells that are believed to undergo very specific autophagic engulfment and degradation. And so the terms have really proliferated here, everything from mitophagy or ribophagy for ribosomes, or lipophagy for lipid droplets, and so forth. But even recognized increasingly is the fact that there could be lysophagy, that is, autophagy directed at lysosomes themselves, which is a relatively new idea, but I think one that deserves a great deal of attention, particularly in lysosomal disease. And again, the issue will come up again a little bit later in this presentation. So now let me move to another sort of general topic here. By the way, I will try to weave all of this together in a few final sort of summary illustrations, but right now I just want to sort of work my way through these various topics and think about how they might be involved in lysosomal disease. And endocytosis, of course, no surprise that it would be involved in lysosomal disease. I mean, we think about the endosomal lysosomal system, and we, of course, recognize today many, many different avenues through which, mechanisms through which endocytic events occur in a variety of different kinds of cells. I mean, ultimately, this endocytic process results in the potential for merger with late endosomes and lysosomes, and so it goes without saying that it's a critical avenue or stream flowing into the lysosomal system. And of course, when you think about the rich activity that goes on within this endosomal system, you know, and you're looking for a way to help explain the complexity of lysosomal diseases, that lysosomal compromise might disturb events within endosomes is certainly in a very attractive way to get at that. Because this endosomal processing can result in termination of signaling, prolongation of signaling, or differences in signaling depending on the location and routing of those early endosomes to late endosomes and so forth. This is further illustrated here in this very nice diagram from Mayer and colleagues, and just showing some of these complex pathways, again focusing on the the early and late recycling pathways leading ultimately to late endosomes and lysosomes. And I would just sort of take a moment out here and just make a statement about another piece of work that we've done in my lab, focused on a condition that we originally called X-linked Angelman syndrome, now bears the name Christensen syndrome, which is a defect in a gene called SLC9A6. The protein is a sodium hydrogen exchanger protein 6. This NHE6 protein is actually resident to recycling endosomes, and yet if you look at our brain paper published in 2011, what you'll see is that this endosomal disease actually has characteristics of a lysosomal disease and affecting many brain areas, including the amygdala, the hippocampus, as well as the cerebellum. And it's intriguing that a protein that's believed located only in the endosomal system, in fact, can cause, at least in many cell types, a lysosomal-like disease. Just again, showing the importance of us in this field focusing on the role of endosomes as well as lysosomes in these diseases. And, you know, again, I showed this illustration earlier from MPS 7 disease, the GM2 and GM3 being in separate compartments. Again, one way to get at that kind of possible separation is, again, to appreciate the complexity of this early endosomal pathway relative to the presence of where the lysosomal enzymes are and so forth. Some of which, by the way, are being trafficked through early endosomes after being taken up from outside of the cell. The other aspects of endocytosis that really deserve our attention come in with particular elements that are within the plasmalemma that are being endocytosed and processed and ultimately involving the lysosomal system. This includes, in terms of, again, focusing on neurons, includes AMPA receptors, the key glutamatergic type receptor involved in excitatory neurotransmission. The lysosomal system is absolutely critical in the cycling, ultimate cycling and degradation of these receptors. Very similar for the receptor tyrosine kinases like TRAC B, TRAC A, and so forth, and the fact that they are routed through the early endosomal system and ultimately into lysosomes. And, you know, I cite these two particular examples really as a lead-in to the next topic that I want to discuss here, one that Herz and Van Hoof and other investigators back in the 1960s and 70s were completely unaware of, but it's this phenomenon that's called ectopic dendritogenesis. And again, those of you who know me know it's a topic of great interest to me, and one that we've known about actually for a long time. It was discovered by Dominick Purpura in the mid to late 1970s, shown here in one of his early Nature papers. What he's showing is a cortical pyramidal neuron from a child that actually had the AB variant form of GM2 gangliosidosis, which means that the enzyme itself was the beta-hexosaminidase enzyme components were not defective. Rather, it was the so-called activator protein, the GM2 activator, which we know now is what generates this AB variant form, was what was defective. So this child was tested for Tay-Sachs and Sandhoff disease and was found not to be deficient in beta-hexosaminidase. This was in the 1970s when cortical biopsies were sometimes done on children with unrelenting neurobehavioral decline, which this individual was experiencing. And this neuron in A actually is from the cortical biopsy that was taken. What Purpura discovered was the presence of this axon hillock enlargement that he labeled here with an M and referred to as a meganeurite. Really interestingly, that meganeurite was covered with dendritic-like spines, which he noted. The child succumbed to the disease a couple of years later, and B is from the autopsy. And what it shows is continuing in the expansion of these meganeurite structures and the growth of actual ectopic dendrites out of this meganeurite area on the cell. This is the growth of essentially a new, you could argue, secondary basilar dendritic system on these cells. He documented this, went on to show it in Tay-Sachs disease as well, not just in the AB variant form, and to show a lot of the variety of different ways that the neurons were being distorted by the formation of the meganeurite and the growth of these ectopic dendrites, which, by the way, are largely separate phenomena in that you can also have neurons with axon hillocks that simply have ectopic dendrites growing out of them, as well as other neurons that may have meganeurites that don't have ectopic dendrites per se. It's really interesting to me. I began working with Dr. Purpura during these days, and my own just fascination with this phenomenon and thinking surely someone else has seen it previously. Indeed, the very notable neuropathologist Max Bielschowski in Berlin at the time had, using a silver stain, had in fact illustrated the presence of these neurites growing out of an expanded axon hillock back in 1921, showing that cortical pyramidal neurons had this kind of feature, but other motor neurons did not, which was another finding that Purpura had focused on as well. Ectopic dendrites don't grow out of just any neuron, and the cortical pyramidal neuron is the key one where they do. And so over the last years, we've learned a great deal about this process. We know that it's unique to lysosomal diseases, but really it's not confined to primary GM2 gangliosidosis by any means, even though when we find ectopic dendrites, what we discover is in fact that they occur on neurons that do contain lysosomal accumulation of gangliosides. And again, going back to that feline model of alpha-melanocytosis I mentioned earlier, this was in fact what we found in that disease, such that those neurons that had the PAS positive material were the ones that were growing these ectopic dendrites out of the axon hillock, whereas the neurons undergoing non-ganglioside accumulation did not. And again, we saw this both with EM and with antibodies to GM2 ganglioside. So there's some kind of link between the endosomal lysosomal processing of gangliosides and misprocessing of those glycosphingolipids, and then the type of neuron. Again, as I mentioned, it's not, ectopic dendrites don't occur on every kind of neuron, but predominate on, for example, cortical pyramidal neurons. And we know this process occurs only after normal dendritogenesis is complete. We know from studies, again, my lab has published much of this work. Happy to send you references if you're interested. We know that it's probably these ectopic dendrites and their new synaptic connections are not reversible with treatment, which is a really interesting thought in its own right in terms of therapy for diseases after these processes are formed. We know that these ectopic dendrites occur in humans. We know they occur in cats. They occur in dogs and sheep. Unfortunately, and it's been a major obstacle in terms of further study of the mechanism underlying ectopic dendritogenesis, they really don't occur to any great degree in mouse models. But what they do suggest in terms of mechanism is that some kind of signaling changes within the endosomal system, likely involving growth factors and neurotransmitter receptors, both of which have influence on dendritic plasticity, could in fact be the trigger for this remarkable event. It's also fascinating to me that if you look during normal early brain development, in this case looking at the development of the cerebral cortex in cats, then in fact the same kind of glycosphingolipid processing within endosomal lysosomal organelles within cells can also be seen. This is not a GM2 gangliosidosis. These are normal neurons showing the presence of gangliosides within compartments within the cell during what is this period of normal dendritic initiation and outgrowth in immature neurons. So now let me move on and say a few words about neuroaxonal dystrophy, another major cellular abnormality in many lysosomal diseases, and again, one that honestly I think has not garnered the attention of investigators in the field to the extent that it should. We still do not understand why this type of axonal abnormality occurs in lysosomal disease. This is from a paper that, again, that Matt McChaney in my lab published a number of years ago, working with the ML4 mouse model with Sue Schlagenhaupt and colleagues. And what's evident here on these Purkinje cells is the formation of these enlargements along the axon, which are filled with sort of a mixture of different organellar materials that are strikingly different than what one sees back up in the cell body. This is a theme that plays out time and time again. The kind of material that accumulates in a spheroid is more generic and not unique like what you find in terms of the ultrastructure of the storage material back in the cell body. Again, just another illustration, in this case a GABAergic neuron that was labeled with horseradish peroxidase with an intracellular probe, just showing the presence of the spheroid in this case a GABAergic neuron in the cerebral cortex. Of course, Purkinje cells are GABAergic neurons within the cerebellum, and again you see the makeup of the spheroid somewhat resembling what's in ML4, and again this is GM1 gangliosidosis that would contain membranous cytoplasmic bodies back in the cell body. So, and there are no membranous cytoplasmic bodies down in this axonal spheroid. And so it's been, interesting to think about this process and to try to characterize it in more detail. We've done this through looking at a number of different lysosomal disease models. Here again, returning to alpha-manocytosis and looking at using looking at the cerebellar white matter and using an antibody to glutamic acid decarboxylase, the synthetic enzyme for GABA, which of course is the neurotransmitter used by this enzyme. And it just so happens this antibody labels axons nicely, particularly in the disease state, and labels the spheroids really well. And you might look at this structure at low magnification and think it's a terminal end bulb of a degenerating Purkinje cell axon, but looking at it at higher power, what you see is that, we see the GAD accumulation within the spheroid, but the axon is continuing. And so the GAD is just one of the elements that is getting caught up in that structure. But the axon is not degenerating at that level. It's intact and continues on down to the deep cerebellar nuclei. The other thing that's impressive here is the size of the spheroid. They really can be quite large, and they're large enough really to have all sorts of physiological consequences on action potential propagation. And in fact, given their size and their frequency in many lysosomal diseases, I've speculated years ago that they're probably one of the dominant factors generating neurological disease in the face of surviving neurons over time, and yet they're really very, for the most part, not being studied sufficiently by those of us in the field. One other point, there was a lot of confusion early on about the difference between spheroids and meganeurites in the earlier literature, and just to point out here, by showing these two illustrations, one, the meganeurite now from Niemann-Pick C disease with its ectopic dendrites, the initial segment of the axon is getting pushed distally as that meganeurite grows. And you can see that here with the with a histochemical stain for the sodium channel-rich region that marks that initial segment. So the meganeurite is a parasomatic axon hillock component or structure. The spheroids, in fact, are axonal in nature and occurring much further down the axon. So they're really completely different structures. The meganeurite also contains the same storage material that the cell body accumulates. So why does this happen to axons? We know that the lysosomal system is largely resonant to the cell body of neurons. It's not present in axons per se. At least that's the view that most people would express. And so you've got an event, a lysosomal disease event occurring in the cell body, and yet far distant down the axon, you've got this compromise going on and an axonal spheroid, which to a very large degree looks like a block and retrograde transport that is occurring on those neurons. So what is the link between the lysosomal defect in the cell body and the potential retrograde axonal transport defect, axoplasmic transport defect down in the axon? And the truth is we don't have an answer to that question. My lab has also speculated that spheroids, as if they are blocking retrograde transport, may be blocking transport of growth factors up from the axon terminal, and in fact may ultimately be leading to the death of those neurons. And the fact that Purkinje cells and Purkinje cell death is so common in lysosomal diseases may in fact be a simple indicator of that kind of deficiency in a sustaining growth factor reaching the cell body to keep those neurons alive. Again, much more work needs to be done on axonal spheroids. Let me say, move on now and say just a few things about events outside lysosome. It's very, very important to recognize, again, just thinking about the complexity of these diseases, and I'll take Niemann-Pick C disease as an example. If you look at in autopsies of children dying with Niemann-Pick C disease, literally 100% of them will show the presence of a tauopathy, neurofibrillary tangles, but that are really identical to what the paired helical filaments that you see in Alzheimer's disease. So Niemann-Pick C disease is not just a lysosomal disease, it's also a tauopathy. But it goes even further than that. Many cases of Niemann-Pick C also exhibit Lewy bodies and Lewy neurites, as illustrated here, which of course are characteristics of Parkinson's disease. And so there are, in this complex expanding disease cascade of a lysosomal disorder, there aren't just lysosomal abnormalities going on, there are other events outside the lysosome as well. And this topic is going to be covered in terms of lysosomal problems in neurodegenerative disease by Randy Nixon in an upcoming webinar, and it'll be interesting to hear that overview. Again, I won't go further into that here. Let me say a word about secondary hyperactivity of enzymes in lysosomal diseases. This is something that Hares and Van Hoof and all of the individuals contributing to that 1973 text recognized. Those of us, any of us who've ever tried to diagnose a new lysosomal disease and looking for a deficient enzyme know that those enzymes that aren't deficient tend to be elevated in activity. And this has always sort of been assumed to be just a feature of the disease. You've got more lysosomes in those cells, in the organs being analyzed, and so you've got more lysosomal enzyme activity. Well, remarkably, Marco Sedelio and Andrea Bellabio and their colleagues took this one step further, and as published in Science back in 2009, they looked for a transcription factor that in fact could be orchestrating this expression of lysosomal enzymes, one that would be doing this where that expression would be elevated in concert across many different lysosomal enzymes and maybe other lysosomal proteins as well. And they discovered such a transcription factor. I've referred to this as one of the most important discoveries in terms of lysosomal disease since I've been in the field. I think it's just an absolutely remarkable finding. And so it's transcription factor EB or TFEB, which they identified, which is orchestrating not only the expression of lysosomal enzyme genes, but other lysosomal proteins as well, and can increase the number of lysosomes, can also increase autophagy, all through this TFEB-stimulated mechanism. So something like 500 different genes have been recognized as targets of TFEB, and again, with really multiple players involving the lysosomal system as being involved, including lysosomal biogenesis and autophagy, as well as stimulating TFEB being a stimulator of lysosomal exocytosis. And let me just say one word about this. I think it's a topic of great interest and one of emerging importance in the cancer field and elsewhere. And for those of us interested in the brain, I think one as well with a lot of intriguing questions in terms of stimulating lysosomal exocytosis, for example, as a means to jettison storage of material out of a diseased neuron. And so the questions come up with, can this mechanism of exocytosis where the lysosomal membrane fuses with a plasmalemma, maybe to repair the plasmalemmal membrane, as has been argued, or as a way to jettison the storage material, whether that could be exploited as a therapeutic tool, not just for liver or for non-neuronal cells, but actually in the brain as well. I think it's really a topic of great interest and one where, in terms of, again, the brain, a great deal of more work needs to be done. So finally, let me touch upon this issue of lysosomal rupture and lysosomal membrane permeabilization is the term is now used in a more modern sense. Again, this goes back to the very first slide in this webinar where I was talking about the dude's early idea about the injured lysosome and what it might do. that lysosomal membranes could break down and release cathepsins and other enzymes now is being focused on in a very modern sense, again, in terms of cancer chemotherapy and in other ways. Again, fascinating area that we weren't, in my lab, looking for per se, but recently stumbled across, essentially, as, again, Matt McChaney was looking at late infantile Batten disease, and again, characterizing this accumulation in aggregates of the macroautophagy adapter protein P62, which we found to be accumulating, and yet it was accumulating in a disease where neuron death was known to be a characteristic, but where autophagy, at least on the basis of LC32 elevations, was not really a prominent, didn't appear to be a truly prominent player. And so in looking at the P62 further using immunogold labeling, we found neurons that had the typical curvy linear storage bodies in their cytoplasm surrounded by a membrane, as you'd expect with lysosomal storage of this particular substrate of subunit C of mitochondrial ATP synthase, for example, which is thought to be the major protein accumulating. But then with P62 labeling, we found other collections of curvy linear bodies that were not membrane bound, but that were labeling with P62. We also found organelles that appeared to be membrane bounded in part that seemed to be spilling out their contents into the cytoplasm. Again, the contents being this characteristic curvy linear profile, which is a characteristic of the ultrastructure of the storage in this disease. In looking at this further, what Matt McChaney was able to show was through confocal microscopy that there were areas of the C subunit storage that were lying outside of the area of lamp staining, indicating a non-late endosomal lysosomal localization. And in fact, these were the areas where P62 seemed to be reacting, suggesting that P62 was in fact reacting to the spillage of storage material out of ruptured lysosomes. In other cases, the P62 seemed to be decorating the outside of LAMP2 positive structures or these enlarged lysosomes as if they were possibly in some way maybe protecting the integrity of that lysosomal membrane, or as I mentioned earlier, perhaps preparing that lysosome for a lysophagy event itself. Clearly an area that needs a greater study. So we published this recently in the Journal of Neuroscience, and again with the idea that lysosomal membrane permeability may be playing a role in the pathogenesis of CLN2 disease, and there are publications by others in other lysosomal diseases that have suggested the same thing. I think it's an area where we need to focus additional work. So I've gone through my list here, but I'm going to add one final topic, again, an area that could not have been foreseen by the pioneers who contributed to that 1973 text, and that's this emerging story as the lysosome as an organelle that's acting as a nutrient sensor in cells. I think, again, this is one of the most exciting new developments in the lysosomal field in modern times. And it really grows out of work with this really master regulator of metabolism, the mammalian or mechanistic target of rapamycin, discovered by David Sabatini, and worked on as well in relation to TFEB by Andrea Bellabio's group, and with really a series of remarkable publications from these investigators. What we know now, of course, is that mTOR forms these two complexes, and mTOR, of course, is known for its ability to regulate a whole variety of cellular functions involved in growth and metabolism. And there are these two complexes that have emerged, and the mTOR complex 1 in particular I would focus on here, because it's known to have close connections with the lysosomal membrane surface, where it's activated by growth factors as well as nutrients, for example, amino acids. And in its activated state, what we know now that this complex not only promotes protein synthesis and lipid biogenesis and overall cell metabolism, but it's doing this through a variety of transcription factors, and one of them is, in fact, TFAB. And as the mTOR complex 1 phosphorylates TFAB, it retains TFAB within the cytoplasm of the cell. And it's really when, in starvation conditions, when the mTOR complex 1 is not activated, that TFAB is not phosphorylated, and non-phosphorylated TFAB can build up in the cytoplasm, can travel to the nucleus of the cell, where it then can orchestrate this expansion of the lysosomal system and autophagy in the way that I alluded to earlier in the papers by Balavio and his colleagues. So this then emerges as really, the importance here is it puts the lysosome really at the center of cell metabolism in a very special way. And some of you have heard me talk about the lysosome as the center of the cell universe and so forth. My way of just trying to get away from the textbook image of a lysosome as this cul-de-sac organelle where things go in, but nothing is ever shown coming out, and no relationship to cell metabolism is being shown. This story has to change in our textbooks now, because the lysosome really is a central player in the metabolism of cells, and we can think about that in part from its involvement in endocytic mechanisms, as well as in terms of autophagic mechanisms, whether it's macroautophagy or cell-mediated autophagy or microautophagy. The lysosome is central to all of that. And of course, what goes into the lysosome has to come out in some way. Lysosomal salvage of precursor materials is critical to the operation of the lysosome. In fact, that's what you see in a lysosomal disease, is a sequestration of material that's not being degraded that is potentially depriving the cell of materials that are critical for precursors to a variety of synthetic pathways. We've argued this in terms of the ganglioside area. Others have argued it for proteoglycan synthesis. I think it's another area that needs addressing. And of course, you put this together, then the better way to think about this is the lysosome as the greater lysosomal system, with lysosomes really acting in concert with the endocytic stream, with an autophagic stream, really, and with this salvage stream coming together to maintain cellular homeostasis. And of course now we know doing this through this remarkable mTOR complex 1 and its relationship to TFAB. And no doubt other players still to be discovered. And of course, if you're looking at neurons, you have to put this together, these events occurring primarily in the perikaryon of the cell body of the neuron. You have to put it together with trafficking out into the dendrites. You have to think about it in relation to autophagy streams within the cell body of the neuron. But you also have to think about those retrograde and anterograde elements that are occurring in the axon, because the lysosomal system is largely resident back to the cell body. So I'll leave you with that, and with this notion that the lysosomal system is indeed a very complex system. And if you want to understand or think about why a lysosomal disease is so complex, you don't need to think any further than, in fact, the importance of those lysosomes and the roles that they play in terms of the overall metabolism and health of individual cells, particularly cells that are with us for our entire lifetime, like our neurons, where these mechanisms have to work in exquisite fashion. So I I really want to thank my colleagues whose illustrations I've used here, and I apologize if there were names I didn't mention along the way. It's a vibrant field with many talented investigators, and I'm indebted to the work they do and the insights they generated. I'm also indebted to a large number of individuals on this call and elsewhere who provided genetically engineered mice to my lab that we have used in the studies that I reported here. And of course I want to thank the members of my lab and their many contributions. I mentioned Matt McChaney a number of times, who has been with me or was with me for a number of years, has now moved on. But there were many others as well that I've not mentioned, and my apologies for that. The NIH has been critical for the support of this work, as well as patient support groups that have been incredibly generous in supporting particular projects involving different lysosomal diseases, and I'm truly grateful to that support. And finally, I just want to thank the families and the individuals who are affected by lysosomal diseases for their courage and inspiration. And for those of you listening to this, and if you haven't gone and met children who have the diseases that you're studying in the laboratory, I strongly encourage you to do that, because it will embolden your work and inspire you to do more on a daily basis. So thank you, and if there are those who would like to engage in some questions now, the hour is late, but I'm happy to take questions.
Thank you very much, Steve, for that great webinar. And I would encourage anyone who wants to ask a question to do so through the question box on your control panel.
I'm sorry, say that again?
If anyone has questions, just go ahead and put them into the question box on the control panel.
So, Mary Beth, are you still with me?
This is actually Siobhan with you here, Steve.
Oh, Siobhan, okay. So are we maybe there are no questions.
Yeah, it looks like we haven't gotten any on this end. But if people want to email you too, would that be an option?
Oh, absolutely, of course.
Okay, I thought that was a great talk, and thank you again.
Okay, well, thank you.
All right, thanks so much. Take care.
Bye-bye.
Bye-bye.